something like this because I remember that during my undergrad, I was so excited to go to grad school. I started my master's, I was working in a lab, the techniques were great, the science was fantastic. But I also felt like there's this emerging field of metagenomics that I barely learned about when I was undergrad. And now as a master's student, there was other people doing it and I had no idea what it was about. So after I finished my master's, I made sure to switch fields to get into this field because I knew it was the future. So here, meta-omics is still the future and still thriving, but we're building upon it. Okay, so I love the introduction. We talked about a metagenome and what that is. So to start off, just as a recap, a metagenome is just taking nucleic acid, DNA specifically, from a sample, and you're reconstructing all of the genomes in that sample. So this is everything that's in that sample. So this first figure I wanted to show you is from Simon Rue's 2019 paper, where he's showing the rise of viral populations. So let me orient your view here. So on the y-axis, we have number of sequences, and on the x-axis, we have a timeline. So the very first metagenome was about 2005, and this was done in Jill Banfield's lab with Gene Tyson as the lead author, where they were looking at a metagenome from an acid mine drainage. And then arguably the first virome was about 2007, but our techniques were very different than they are now. And the main thing you want to take away from here is that in green and blue, we have everything we know about viruses from isolates. And blue meaning we have all of them, and then in green meaning these are complete genomes. So even in isolates, we don't always get complete genomes. And then with the advent of metagenomics, the discovery of viruses has exploded. And you'll see acronyms out there like UVI or UVIGS and da-da-da. These just mean viral populations. So when we talk about viruses, there's so many nuances in their genomes, like you're probably imagining with COVID going around now, the SARS-CoV-2, there's all these different variants, right? They can be described into populations, especially if we're trying to understand how the community is interacting. So the way we do this is we take similar organisms and we cluster them based on a sequence similarity. So we'll typically use a 95% average nucleotide similarity, and then we'll do a cutoff for the genome length. And we typically are looking at double-stranded DNA viruses for most metagenomic studies. So what we'll do is we'll usually do a 10 kb cutoff. So we'll say 95% average nucleotide identity across a range of the genome. And the currently accepted threshold is 85% of the contig length. So we cluster these genomes together, and the longest one that they cluster in is our representative viral population. Now you can imagine we're going to missettle these and differences amongst these genomes. So it's always good to dive back in and really look for differences between these viral genomes, especially for gene specificities. So just moving forward, this is where the field is now, is that we're getting this vast discovery of viruses from metagenomics. Metagenomics is our main tool. And metagenomics is really good at capturing double-stranded DNA viruses because the technology was set up for microbial genomes, which is double-stranded DNA. And from a metagenome, you can get the double-stranded DNA viruses, and you can also get the microbial host information, so you're able to link them together, which is great. Now, a problem is we get low resolution on viruses, which is what the remainder of my talk is going to be about. To show you how things have changed, myself and others have really been pushing hard in soils because they've languished. So on the figure on the right here, we have the number of virus-specific meta-studies, and the total has exponentially increased. And it seems that human and marine are the ones taking off the most, but we're trying to push to get soils up there as well. And then there's so many other fields in between that are just, I didn't bring up. Okay, so let's discuss the problem. We know that you get low resolution on viruses. We get on average about less than 2% of our assembled reads going to viral contigs. And it seems like the most of these viruses are described are viruses that are either integrated or associated with the most abundant hosts. So we're bottlenecking and obscuring our view to only focus on these most abundant ones proxy to their hosts. So the reason why we get such low resolution on viruses in general is just because if you look at a soil sample, there's so much going on. You're collecting genomic information on plants, on the macrofauna. We're talking about your mites. You can go to your mesofauna. We're talking about your nematodes, and then you can go lower into your microeukaryotes, your amoebas, and then you finally get lower into our microbes, in which we have to zoom in. And this is what a lot of people care about. But then if you want to know viruses, you have to zoom in even further to finally see the viruses. So my whole PhD and what my career has been about is increasing our resolution on viruses. And again, this has application across all fields. Currently, we're wanting to learn how to better detect different viruses in the environment and increase our threshold on which we can detect viruses. So all of these have similar applications. I see that I have a couple comments in the chat. I'm just going to wait to the end to answer those unless there's a pressing issue, and then please interrupt me. I'm wondering if we can somehow move this so it's not blocking. Sorry about that. Hold on one second. Okay, I'm sorry. So you're not going to be able to see the titles, but I will give you information on what's going on. So what I've started to do during my postdoc and now as a staff scientist is really to understand and increase our focus on what we actually care about. So when you look at an environmental sample, you do want to know everything's there, everything that is there. You want to know what they're doing. This is your genetic repertoire. You want to know all the capabilities of what's going on. But we also care at each point in time, who's actually doing what. And the more we study microbes and other organisms and how they interact, the more we understand that not everything's active at a single time point. That actually in soils, a majority of microorganisms are inactive. They can be active, they can be growing, but a lot of them are also dormant or they're just deceased. And we're picking up what's called this relic or ancient DNA from the soil environments. So to get around this, we use stable isotope probing as one way. So this is where we take an isotope, and this first example I'm going to tell you, it's going to be for oxygen. So we have regular water, which is typically H216O. But we can make what's called heavy water, which is H218O. Not to confuse with deuterium, but we're specifically using 18O. So when we add this to an environment like soil, water is a universal substrate. So anything that's active is going to have to use this water. So it becomes labeled with the 18O within its DNA. So we can detect everything that's active. Now for viruses, if they're infecting a host, then they will also get labeled. So the viruses get labeled as well. In another example, I'm going to show you how we can look at substrate specificity. So here you have plant biomass, which usually has normal carbon, which is 12C. We can grow plant biomass with under 13 CO2, so it's enriched plant biomass. So the majority of all of the plant is 13C enriched. We can then look at who's degrading that plant biomass, because any organisms that are breaking it down will become 13C enriched, and so will their viruses. So this won't label all active organisms and biological entities. This will only label the ones that are involved in breaking down the plant biomass. So it's substrate specific. So what we typically do from this is we incubate the sample. This will be soil for a period of time. Then we extract all the DNA like you would for a metagenome. But then we take the DNA and we put it on a cesium chloride density gradient and we do ultracentrifugation. The DNA will separate into the different layers of cesium chloride density based on enrichment. So you'll have dormant or deceased ones that didn't take up the 18O or didn't take up the 13C up higher in the lighter fractions. The ones that incorporated the heavier isotope that's more dense will be in the denser fractions. And we can physically separate these and sequence them separately and compare them. Now, there are differences in this. Obviously, there's organisms that are GC rich that can make them more dense. So we end up taking many fractions and comparing those fractions. But it's important to always sequence both the light and the heavy fractions. This allows us to get the genetic repertoire of everything that's going on. Remember, we took a snapshot in time, so it could be that the metabolisms that weren't active had just stopped right before we added it for some reason. So this is where time series also comes in handy. But then it also allows us a control to compare it to see differences in growth and changes in the communities. So the first example I'm going to talk about is with the heavy water, and this is a paper I recently published, I think came out in August of last year. And this is where we went to Bonanza Creek in Alaska in a zone that has permafrost, which is ground that's frozen for 2 or more consecutive years. Now, the assumption is in these northern latitudes, Siberia, Scandinavia, Canada, Alaska, that over the winter, organisms just don't function or it's minimum, so they just ignore a lot of the carbon that leaves these soils and they don't want to measure it. But we've known for quite some time that this is false. So we wanted to actually connect microbes and viruses to carbon cycling over winter months. So we sampled Bonanza Creek and we collected the peat soils. And then we took the soil and we incubated it in a jar for half a year and for a full year. And we did some of the samples with regular water and then some with enriched water. And then we also brought the temperature of the vials down slowly over time to negative 1.5 Celsius. And we also made sure they were anoxic with a nitrogen headspace. So this will mimic the in situ winter conditions. After the incubation, we then did the cesium chloride density fractions and we sequenced them separately and we reconstructed 23 metagenomes. Along with this, we were collecting headspace measurements so we can measure CO2 production over time. So in the figure at the bottom, you can see in purple from our samples that we had CO2 production that occurred over the full year incubation. We also saw in our other control, which we brought down to negative 20, that there was no production. So somewhere in between there, we have our limits of life being able to respire. So I'm going to just do some quick highlights, but the main things I wanted to focus is on the viruses here. So I have a heat map here. On the x-axis, we found 332 viral populations. On the y-axis, I have 4 different treatments. We have regular water for a half a year, enriched water for a half a year, and then both for a full year, and I compared them. So if we look at just the bottom part, that's our active ones. So we found that 243 of our 342 populations were active over the winter. So this is incredible. This is a large amount of viruses active in the winter. If we're looking at just the active ones now, we first noticed, comparing a half year to a full year, that many of the viral populations persisted. So if you look from the half year to full year, they're still there. So that's the first thing to note. The second thing is that a large portion of them actually increased their abundance over time. So it's not like they were active and then they just sat there. They continue to infect or multiply in the host over the rest of the year. And some theories on what could have happened is that these could have been tempered viruses that were integrated in the host, and as the host doubled, the virus also doubled. It could be that they burst out of their host and made virions, and the virions persisted in the environment. And we do get a little bit of this in our metagenomes, but it's not targeted like a virome, so we don't get a lot of virions. It also could be that these viruses and maybe the host died and exploded, and because these are peatland soils and they're in soils, the DNA is protected, so it could be that they just remained as relic DNA in the environment. I also want to highlight to the left, you can see we had viruses, 8 viral populations that were present at a half a year and were no longer present after a full year. So we're seeing this dynamic community in which populations are leaving and populations are coming into play. So then that brings us into the 151 new populations that just occurred from a half year to a full year. And not to discuss too much more about it, but if you look at the host, it seems like the viruses are mimicking the host, that when the host populations are changing, the viral populations are changing too, following the kill the winner hypothesis. To talk about the host a little bit, we had this huge diversity where we had 10 bacterial phyla in these sub-freezing anoxic soils, which is incredible in itself. But we also saw that 7 of the 10 phyla were active. So we had bacteria that are active over winter. So that means they're doing something. And if you were to take a traditional metagenome, you would have sampled all of these, and you would have seen that there was 10 bacterophyla. You would have also specifically focused on actinobacteria, because if you look at general read information and coverage, the actinobacteria dominated the signal. But this just occluded the actual truth of what was going on. When we actually looked at the active organisms, we saw that actinobacteria were not even that active, that the majority of them were actually firmicutes and bacteroidetes. So we would have missed this signal had we not done the stable isotope probing metagenome. Of those 7, only 4 actually had viruses in which we detected were infecting the host. And then they changed throughout the year as well. So I wanted to highlight that we had the Patsy bacteria. These are incredible because these are ultra small bacteria, part of the candidate phylum Radiation, which is newly discovered. So these organisms are interesting because they don't really have a free living system. They have to connect with other hosts. So they typically can connect with bacteroidetes or actinobacteria. So there may be some syntrophic relationship with viruses infecting multiple hosts. We also found our proteobacteria, specifically these vampiric bacteria, which these are bacteria that have to attach to other bacteria and feed on them to survive. And we found viruses infecting these bacteria. And then again, as I said before, the firmicutes and the bacteroides were the most abundant phyla. And these are the ones the viruses switched to infect throughout the year. Okay, so just to sum it up, I'm happy to talk more if people want to know more about the paper, but some key takeaways. There's major differences between the active and the unlabeled communities. So there is promise in trying to decrease our complexity and look at the active portion only. We found only a few bacterial populations that were active, but we found a ton of viral populations that were active. When we looked more to the host abilities, they had the ability for fermentation and to also break down carbon, and they produced CO2 throughout the whole sample. So this is important because this showed and links bacteria to producing carbon dioxide over winter processes. So we need to be thinking about CO2 loss over the winter, especially because there's some studies out there that suggest that CO2 loss over the winter is actually more than the entire plant uptake during the growing season. So we had 1/3 of our viruses were linked to active hosts. We may be able to increase this if we were able to do a viral targeted metagenome, also known as a virome. We also found that viruses included these key genes to modulate the host metabolism. So these are known as auxiliary metabolic genes. These are genes that are outside the virus's normal repertoire to infect the host, and sometimes can bring genetic novelty to the host or can modulate the host metabolism. In this case, they help the host with carbon degradation and also scavenging nutrients from the environment, probably aiding in survival. And then I think it's important to note that the viral community did shift from who they infected from a half year to a full year, which was supporting the kill the winner hypothesis, showing that the viral communities do shift to who the most abundant host is. Okay, so for our next experiment, this is going into our 13C enriched plant biomass. We're now shifting gears, getting out of that frigid, cold, Arctic environment. We're now in the lush tropical rainforest in Puerto Rico, the Luquillo experimental forest. So this is, Puerto Rico is part of the United States, and this is the only tropical rainforest that is part of the United States. So we sampled there. And what's important to know about these environments is they have tropical storms. So it's a common occurrence for there to be heavy rainfalls and dry periods. So these soils undergo heavy redox conditions where it can be anoxic and then oxic. And we're wondering how this affects the fate of the carbon and what tools and traits have the microbes picked up to survive, and what are the viruses doing? So this experiment was designed by Ashley Campbell and managed by our group leader, Jennifer Petridge, to the far right. And then I had Jeff Kimbrell and Steve Blazewicz that helped me as well. And they designed this experiment where they took soil samples, and for 44 days, they incubated them under 4 different treatments. And they added the 20 grams of the soil with the plant biomass. And I just want to remind you that this is not labeling all active organisms. So we're specifically looking at ones involved in degradation or breaking down the plant biomass. So the treatment setup was, it was either completely oxic with air being flushed in. It was frequency between oxic and anoxic at a high amount or a low amount, or we had ones that were completely anoxic with an N2 headspace. From this, we were able to take 95 metagenomes. So this is huge. We had terabytes of data. Now, we wanted to really compare and ask the question, when we do the CYP fractionation and we're looking at the active ones, are we actually decreasing the complexity enough to actually get increased resolution? So we took 85 CYP fractionated metagenomes. This is where you extract the DNA, and then we do the different fractions to see which ones are heavy and light, and then we compare them. And then we had 10 bulk soil metagenomes. This is just, we took some soil, we extracted DNA, and we sequenced it. So this is your traditional metagenome, and we wanted to compare them. Okay, so I have a heat map here again, and let me orient you to how it works. On the x-axis, we have our viral populations. You can see already we had double the amount of viral populations compared to our Arctic samples. Now for the color scheme, an orange means, white means not present, orange means low abundance, green means high abundance. And then on the y-axis, I have it separated from, in black, our bulk metagenomes with our different treatments. In blue, we have our no additions. This is just plant biomass, so it's our control. And then in red at the bottom, we have our 13C enriched plant biomass that we added to compare. And you can see, we see viruses in all the samples, and there are some changes. So now we're going to dig more into those active ones, the ones involved in carbon degradation. So looking at this, the first thing you should notice is that there's more viral populations in the oxic samples. So we've known for quite some time that oxygen really affects viruses. And it makes sense. When a virus is infecting a host, if it's lytic, it wants to take over the host and make progeny viruses. This is oxygenic metabolism that you need. So we see about a third of our viral populations were here. And then you see some changes when we have redox conditions. But what is super cool, so we have about a third that are in all the conditions. But what's important to note is that in the oxic ones, we have 27% more viral populations. So you think oxygen is important. But then if you look at the anoxic ones, while we have less diversity, the viral populations that are there are the most abundant. So here I'm thinking we have some viruses that, you know, they probably don't have a broad host range. They probably don't have these large genomes. These are specialists. They've changed the way they infect to change the host metabolism, where now we have this anoxic metabolism, completely different pathways, and viruses evolved to work with this. And we don't have these oxygenated viruses here. So this is a niche spot just for these viruses. And this story is not published yet, so I'm still actively looking into what these viruses are doing, because this has important implications for us, right? We have parts of our body that are oxygenated, and we have parts of our body that's anoxic. So our microbiome, including our viruses, are going to change in those different redox conditions. Okay, so then just to highlight the host real quick, we had massive diversity. I mean, these are tropical rainforest soils. This is something you would expect. We had 20 phyla of bacteria detected. And in here, in this figure I have here, I have a alluvial chart. On the first column, we have all of our viruses. In the middle, I was trying to get an understanding of connecting viruses to the host amongst the different redox treatments. And then the far right column, we have our different phyla of bacterium. The main takeaway I want you to get from here is that we have huge diversity. And if you kind of follow the green, which are the viruses that were 13C labeled, they don't go to all phyla. They specifically span, I think it's 8 phyla. And we wanted to look more into those specific phyla because these are the ones that are really controlling the fate of the carbon in this environment. So the next thing we want to do is wanting to look, well, what are the host capabilities? Because the virus can only do so much for the host they're infecting, right? You're driving a car. Now, if your friend has a Mercedes and you have a little scooter, you're both driving a vehicle, but they have more influence. They have the capacity to go faster than you and further than you because of what they're in. So this is like the virus infecting the host, right? It's limited based on the capacity of the host. So what we did is we wanted to look for the various ways they could break down carbon substrates. So I'm going to show you all of them, but I wanted to zoom in on one to orient you and get you thinking about this. We have the phylum Actinobacteria. On the y-axis, we have the number of genes within their genome. And on the x-axis, it's the number of genes within that substrate class. So the first one's cellulose. So every circle there is a bacterial genome. That's an actinobacteria. And then higher up if it has more genes to break down cellulose. Okay? So higher counts means higher capacity to break down plant biomass. Higher counts across the whole field means a high diversity to break down a range of substrates. Okay? So what we realized is that we took all 20 phyla and we looked at which ones the viruses were infecting. The viruses were infecting the key carbon degraders. So the ones with the largest amount of genes involved and the highest diversity for plant degradation. So the viruses are actually caring a lot about the carbon and influencing the fate of this carbon. So this is just them doing it via infecting the host. By infecting the host, they can increase the host metabolism to make more viruses. They can lie somewhat dormant. I don't like to use the word dormant because while they're integrated, they're just not there. They are still doing maintenance. They're also encoding different toxin genes to help the host and protect the host from other infection and to keep it happy. So there's a lot viruses are doing. But if we look more at other ways the viruses could be influencing metabolism, we start thinking about the different ways in which they can control carbon cycling. So like I said before, they lyse the host. This is releasing nutrients into the environment. That's a top-down control. If we are also expressing AMGs, this is augmenting the host metabolism, we can make their metabolism more efficient and make them outcompete other populations as well. So we looked for these AMGs and we found a whole slew of AMGs within our viral populations connected to our hosts. So we think about this more because I'm not done with this paper yet, but we're looking, what is the fate of this carbon? Okay, so first idea is this kill the winner hypothesis, right? The viruses are shifting who they're infecting. It's hard to see that here because it's a 44-day incubation and there's not really a time comparison. So we really can't see this, but we know that viruses are infecting the most abundant hosts that are involved in plant biomass degradation. So we do see that. So this could be maintaining the microbial diversity, and they're also killing all these hosts, creating diverse substrates feeding other organisms. Another thing you could be doing is the viral shunt, where they're killing the hosts. This necromass, microbial necromass, all the nutrients, is becoming available, but then it's sticking. And in these soils, we have phosphorus limitation, and then we have all these different pore sizes that microbes can't fit in. So it may be that the viruses are killing these hosts, these nutrients are going in these small pore sizes, and it's inaccessible to other microbes. So it just keeps carbon in the soil. And then this leads us to entombment. So by keeping that carbon in the soil and it's inaccessible, it can persist for long periods of time until maybe we till the soil and making it available for other organisms. There's a flood or there's a storm that moves the soil, or there's small organisms like ultra small bacteria that can make their way within these pores and break down this carbon. Okay, now that is stuff that I was currently working on. About a year ago, I got a grant. So this is what's called my LDRD. And this is to really push the field. The government gave me a grant for something that's called high risk, high reward. You're doing science that has a high chance to fail, but if you were able to complete it, then it would be very valuable to the community. So what I wanted to do was other ways we could tackle increasing our detection of microbes and viruses and getting better resolution. And again, going at soils is a great way because there's all of this going on. So how do we tease it apart and really figure out what's going on? Well, we set up doing metagenomes incubated with heavy water. So I did it over a time series. I took some soil, I did 4 different treatments over a time series. So the new component says I'm adding time to really see how viruses and microbes are changing and how their host interactions are changing. But we're also adding in our heavy water to look at the active community over time. But I also added in a phosphorus addition because I wanted to see, these soils are limited in phosphorus. So if we're adding phosphorus, is this going to spur infection? So that was the first part. But that alone doesn't just help us with our detection. So at the same time with these soils, I also took a virome, which is a viral targeted metagenome. And this is actually what I spent my whole PhD optimizing. This is where you can take a sample. For here, I'll take a soil sample. And you add a resuspension buffer to help desorb the viruses from the environment. You physically separate them and you run it through a filter. So now you're looking at the viral size fraction. And then you want to purify it so that way it's not sticking to anything. And then you extract the DNA and concentrate it. So now we're looking at not just the double-stranded DNA that you get from a metagenome, but we've also optimized it so we can get single and double-stranded DNA viruses from the soil. And I've been recently working with JGI, so we have Emily and Simon Rue here, in which we're trying to get stable isotope probing viromes, the first of their kind. So this would be looking at active viruses, but in the virion stage in the environment. So there's no incorporation to the host. Okay? So the benefits here is we get increased resolution on our viruses, and we've shown this with previous work. You can also get increased resolution on the ultra small microbes because they make it to the fraction. But we lose our host information, so it's good to have it paired with a metagenome, which is what I've done. But we're still not done. I wanted to increase resolution of all of our viruses, correct? Then we also added a metatranscriptome. RNA viruses are super important. In a lot of mammals, especially humans and gorillas, it seems to dominate the literature. Like we only care about RNA viruses. But it's funny because for ecology, when you go into environmental systems like soil or water, they seem to be missed out on. We are only looking at double-stranded DNA viruses. But there have been recent studies looking at RNA viruses. So I wanted to be able to capture these also from this same sample. So we're going to get double-stranded, single-stranded DNA viruses, and also RNA viruses. So this will capture all of our RNA viruses. We'll also be able to get gene expression for a host and for our DNA viruses, which would be great. We're still limited to the point where we have to remove tRNA and rRNA because they swamp the signal. So we do lose a little bit of information there. Finally, something that I newly invented and I'm working on is what I call the EDNA Metagenome. There's been recent studies that I think the first paper that really pushed it into the eyes of people was Paul Carini's 2016 paper, where he sampled some soils and they found a lot of eDNA. And that by looking at the eDNA, you're actually seeing variances in the abundances of your hosts. So you need to be careful of how much of the eDNA is actually just relic DNA lying around. And just to clarify, relic DNA, ancient DNA, eDNA, which can be exogenous DNA or environmental DNA, it's all very similar. It's just DNA that's free in the environment that we're trying to understand. Now, I was working with this because there's recent papers from wastewater treatment facilities in aquatic environments showing that if you look at the eDNA, you can actually get increased detection of viruses. So I'm wanting to monitor the viruses and get this increased detection. So we sampled eDNA from our soils too and reconstructed that. So just from this one soil sample over this time series with these 4 different treatments, we have a ton of SIP metagenomes. We have bulk metagenomes, SIP viromes, bulk metagenomes, and eDNA-omes that we're comparing. And this is the work that I'm actively doing right now. So I think in 6 months to a year, start seeing some results coming from this and looking at the whole soil virosphere of a sample. And this is really looking at the virome, really, of the soil, like all the different components of the virosphere. Okay, so I'm going to wrap up now, but I wanted to also talk about another component to make sure that we're not missing out on it. One of the issues with metagenomics is that everyone thinks meta-omics studies, which is metagenomics, metatranscriptomics, metaproteomics, and so on, and metametabolomics, that we're thinking and we can just do it and we're done. We need to make sure that we're taking controls and trying to be quantitative. By adding in stable isotope probing, we can start looking at activity. You can also add in BondCat, which is bioorthogonal non-canonical amino acid tagging, and this can also get at activity. But another way too is we can try to use microscopy to make sure we're quantitative. So what we're doing here is we're taking our soil samples in which we've taken these metagenomes and so on, and we're separating them, and we're trying to count the number of viral particles. Now, epifluorescence microscopy doesn't do so well, especially in soils. So we'll use transmission electron microscopy. But we've also taken a step further and we've paired it with nanosims, which is nanoscale secondary ion mass spectroscopy. And the way this works is you take a sample, you sputter it with a cesium beam, it destroys the sample, but every time it hits a sample, it charges the sample, and the computer can read it and can reconstruct an image. So these figures you see here, these micrographs that are gray, those are TEM images that we can see viruses. And you can see in the top right here, we have M13 phage, which is a filamentous phage that infects E. coli. We also have down here, we've been working with lambda phage, which is an iconic Cyphoviridae shape, and that also can infect bacteria. Now you can look over to the far right, we can start to do nanosims, and nanosims is great because it allows us to not only quantitatively count the viral particles, but we can also see the elemental composition. We can see if it's 13C enriched versus 12C enriched. We can see if there's carbon in it. We can see if there's oxygen in it. So we can look at where the enrichment's going. We can also know what nutrients they're taking from the host and what nutrients they're putting in their environment. So this is something we're actively working on as well to try to quantify phage from the environment, but also better understand their nutrients that they hold on to. Okay, so hopefully that wasn't too much. I'm sorry, I was trying not to go too fast, but I want to leave time for questions. I want to acknowledge everyone I worked with. I love universities. I never thought I'd work in a national lab. Since coming to a national lab, I see so many benefits, how you can really hone in on your science, and I can really connect with universities so I can still teach if I want to and give back to the community. So I really want to acknowledge Jennifer Petridge and Steve Blazewicz. They've been excellent mentors and helped me convert to a staff scientist. We have tons of funding. We have LDRD for high risk and rewards research to Steve that I came in on. I currently have mine as well. We were funded by other organizations for some of the studies. We also have our large SFA, which is a science focus area that's ongoing for the next couple of years, where we're really trying to understand how precipitation affects microbial populations in soils. Specifically in California, we will have absolutely no rainfall really from about May to October sometimes. And so we have all of these organisms, including the grass, that just go dormant. And then from about November-ish to April, May, we'll start to get rainfall and these organisms will come alive. And this really controls the carbon output from the carbon output from the soil. So we're trying to understand how moisture really dictates these communities and their traits. And then lastly, I want to do a land acknowledgement. We're only here because of the people that took care of this land. So I'm super appreciative of being able to sample these lands and work on these lands and have the privilege of continuing to do my science. So with that, I'll just take any questions now.
Thank you so much, Dr. Gary. It was so amazing and so intriguing to learn about these hidden communities of viruses and bacteria, and really their properties of them being dormant and active and shifting and processing the carbon, affecting the metabolisms of the host is so much fascinating. Thank you so much for sharing this amazing presentation and research discussion with us. Well, we have a lot of questions in the chat from Iman, so they are very interested and very intrigued. So maybe I'll put some of the questions for you, which would seem to be most interesting. So this is something which I think many of us heard for the first time, that is the eDNA. So there's a question in the chat that where do you find the eDNA? Where is it? Or is it similar to the multivesicular nucleic acid in liquid biopsy that we find in human beings? So could you explain eDNA once again?
Yeah, so it's an all-encompassing term, and it has so many different ways in which it's said, and they try to mean different things. So at its base, eDNA is either extra polymetric DNA, exogenous DNA, environmental DNA. This is DNA that was in a biological entity. So I say that because viruses are not considered organisms. So whether it's from a virus or a microbe, an amoeba, a human, it's in the environment. It doesn't matter if it's put there on purpose or by accident. So we know that microbes can exude DNA and they can use it to help build biofilms. The eDNA is a huge part of biofilms. We also know when a microbe dies, its DNA goes into the environment. It's sticky. It sticks places. Now, in some environments, it can be degraded rather quickly. If it lands on metal, sometimes in the ocean, there's not a lot of eDNA depending where you are. But in soils, it gets into the various pores and it attaches to the colloids in different ways. So whether it's silt, clay, or sand, it can hide the DNA and it can help preserve it. So peatlands are so popular because these are found in northern latitudes generally. But this is where microbial degradation is limited, usually because of temperature. So you get this buildup of plant biomass, sometimes animal biomass as well. If anyone's from a northern northern latitude, you see this. It's kind of cushy to walk across. And we call it peat because there's not that much soil in it, even though it is soil. It's mainly this plant. And you can preserve things, especially if it's a bog because of the low pH. So we find a ton of DNA just in the environment intact. Now, a lot of times it's highly degraded, so it can be really hard to get information from it, but sometimes it's not so degraded. And this can be in any environment. So in your gut, we're going to find DNA that's just exogenous, so just outside, just free-floating DNA. You're going to find it on your skin everywhere. The push is to really have the capability to measure it and see what we get from it. And for viruses, specifically RNA viruses, I think there's a greater importance because we know for RNA viruses that they can become infectious and be stable as free nucleic acids. So you can have RNA in the environment, and if you take it up, whether you're a microbe, let's say a fungus. So RNA viruses typically are free-floating as RNA in the cytoplasm, and they can remain infectious. So if they're in the environment and they get taken up into the host, they can infect the host. So this presents security questions that we have to think about, or whether it's for crop health, human health, or whatever. So this is definitely a growing field, and I wanted to really step my foot in there. So I'm bringing this up so that all of you as people going into the field know, while meta-omics is the way to go, there's changes as a community where we're trying to become more quantitative. We're trying to see all the different components and tie things together with process measurements to really understand what's going on. So I hope that answers everything about eDNA. I guess the last thing I'll say is sometimes you'll see it called relic DNA or ancient DNA, and these can have different meanings. The relic DNA was originally meant because it's thought that it's DNA that left an organism and it's hanging around for a long period of time, whether it be weeks, months, years, and it's highly degraded. So you don't get a lot of information on it, but you know it's DNA. Ancient DNA is when we're finding DNA that's in an environment that, again, maybe years, decades, and sometimes can be found in permafrost or just in soil. But these generally all mean the same thing. It's just DNA outside of a biological entity.
Well, that's super fascinating and super interesting to learn, and I hope that everyone would definitely want to investigate further into understanding the real role that these things play. So there's another question which I would like to pose you is, so Ilya asks that some people say that the soil microbiome affects tremendously the food metabolites and have a chain effect on the full food cycle. So do you think that it is true, and if so, how? And also, how stable are these communities from season and climate changes, or do they depend upon the time of the year, the time of the day, and so on? So what's your take on that?
Absolutely. I think soil is extremely important because while it's not the largest reservoir of carbon, it's larger than the atmosphere and the biosphere combined. It's huge, but we interact with it the most. We change it the most, right? So if you think of a soil, you can imagine it as a smorgasbord of just nutrients. But when we're growing crops, the crops take out different amount of nutrients. So we're changing the soil. And before we didn't have crop rotation like we did now. So we would heavily deplete soils and we erode soils. We don't treat our soils well. We kick them around. We treat them like dirt. So it causes issues, and our nutrition, which we get from it, right? The plants are just like us. They will work and they'll especially have the help of fungi to really get all their essential nutrients. But you can definitely have plants lacking in nutrients or just plant health where they can be more susceptible to infection in different environments. I think one thing we're going to see here with climate change and a growing population is we're going to see more and more agricultural in northern latitudes, especially in peatlands. I think a lot of them will be drained or changed for agriculture. We know that when you introduce a plant into a new environment, it's susceptible to disease. So this can cause blights, food shortages, other issues. So these are ways that it can affect our security indirectly by not hurting us, but hurting our food. So I think understanding viruses in soil or in plants or in like the phyllosphere, it's all interconnected, and we need to have a better understanding. Our current thoughts, though, are plants are more infected by RNA viruses or single-stranded DNA viruses, but it may be we just don't know, like we're missing a component. So this is part of what led me to think in my peatlands, where I've been studying double-stranded DNA viruses, I wanted to work on methods to get at single-stranded DNA viruses to really see, if there's plant material here and there's more viruses for plants, then there should be single-stranded DNA viruses. So this is stuff we're looking into. And then your last question was something about climate change, about how stable the communities are. So climate change is one of my favorite topics to think about. And, it's hard to stay up to date on the literature. I'll say that Kristen DeAngelis at University of Massachusetts Amherst has a lot of good research on this. She's studying soils under warming temperatures and stability of communities. There's a lot of research at this at Berkeley as well. Look at a lot of the face sites. There's a spruce site in Minnesota. So together, what I think I remember so far is the communities are stable, right? You don't see huge changes. But this is also from us just taking general metagenomes, right? It could be that the community is there, and then this part activates in the summer, and then this part activates in the winter, and then they work together, right? So I think we need to know more of who's actually active when to see who's interacting. I've also spent quite a lot of time studying viruses and microbes with permafrost thaw. And we see that if there are dynamic changes to the soil, we have changing communities. So in northern Sweden, where I've done a ton of research, that's Stordal and Meyer, and an intact permafrost, which was the dominant previously in the Bisco fields in the Bisco area, was this pulsa habitat where it's shrubby, it's kind of bouncy, it's like a temperpedic bed. You have these stable communities. But then once permafrost thaws and you get a little bit of ground collapse, depending on how much water or ice was in the permafrost, then you can get this bog-like environment. And it's hard to go back after that because then when you have a bog, you promote new plant species that come in. So you get sphagnum moss to grow now. And this sphagnum moss, when it comes in, it takes over. It produces this galactochloric acid, which lowers the pH, inhibiting microbial and other plants from growing. So you only have certain bacteria. So in the pulsa, you'll have a diversity of shrubs, and you'll have diversity of bacteria and archaea. When you get into the bog, you now have this increased abundance in acidobacteria because they can tolerate the pH. So you have, boom, it's taking over now. So you've just changed the microbial community and the viral community because the viruses are infecting that host, right? Then you're also changing the plants because now you have the sphagnum moss that's there. And now it can be somewhat flooded and it's going to keep that water there. Well, then and this is partially thawed, then you go into a fully thawed habitat, which in this habitat happens to be the sphen, which is dominated by this Areophrum, which is the sedge. So the sedge comes in, it grows this large structure, it can pump some oxygen down, but now we have what's like a wetland ecosystem, where in the pulsa, you had some methane production, but you also had methanotroph, so it's really respiration we care about. In the bog, you start to have hydroneotrophic methanogenesis. So you start getting various methane coming out and some respiration as well. And then in the fen, you have the switch to this acetoclastic methanogenesis. So a complete change in the metabolic functions and this high abundance of this archaea. So you have this wetland with a lot of methane coming out and a different plant species. But then you also have the diversity of microbes increase again. So it goes from about here to here to here. So we do see a change with climate change. But to give you more specific answers, it depend on the field, the habitat, and we'd have to look into those papers to assess it for that habitat.
Right. That. Thank you so much for such a wonderful explanation and such a detailed answer. And I completely agree that it would depend upon so many factors as to what would be the change for these communities. Well, one thing which was very interesting from your presentation was the integration of various techniques that you brought together, being it metagenomics, viromics, the different aspects of viromics, mass spectrometry, and ultimately microscopy, and also the new form of microscopy that is the nanosims. So, what were some of the data integration challenges that you might have faced while dealing with and working with so many different types of techniques and the data sets generated as a result of those techniques?
Yeah, so there's many different things we could talk about. I think the first thing, which is upsetting, is the amount of specialty and money that's involved, making it not available for everyone. So with the first metagenome about 2005, metagenomics, because of next-gen sequencing, has gotten much cheaper, but it's still thousands of dollars. I've spent nearly $100,000 this past year on various sequencing costs, and not every lab and not everyone can afford that. So there's cheaper ones where we'll do amplicon studies with 16S rRNA gene surveys or 18S or ITS. So these are ways people get around it. So I think a lot of labs are limited financially and with the expertise and availability, so they do the best they can. So it's troubling that we have proposals, we have calls, and we have people propose, and the ones getting it are the ones that have the financial capability and the means to do it, so they get it. And there's not really good calls to say, hey, here's what's needed. There's all these labs that don't have that, so these labs can apply for it so we can bring them up. So there's more labs able to work together to make this science better versus selecting a few. And really, if you're a newer lab, the way you can increase your career is you somehow get involved in a group with one of these other labs. And then it shoots your career up because you have all this financial availability and expertise. So for some of the smaller labs, it's not possible. And so this is upsetting that we're limiting who can do what. So the things you saw in my work, a metagenome, okay, a lot of people can do that now. A lot of times they'll have it paired with amplicon studies. That's good. You can do some qPCR, that's great too. But then to do a metagenome and a metagenomic, this is actually quite challenging, especially if you have to know the expertise on getting this from soil. So we're losing some labs here now, and we're losing the financial capability. And then if you want to add metaproteomics or metabolomics to really see it from the beginning to the end, it's more expensive. And I want to highlight how important that is for everyone. A metagenome tells you the potential, and you can get a catalog. So a metagenome is the base and you need it. The metatranscriptome is going to tell you the expression of what's happening, or if it's for viruses, it can actually detect viruses, but you need the metagenome. And it's not for certain. We have a lot of microbes that will produce mRNA like chunk change to try to get ahead of other microbes. It doesn't mean they're actually doing that. Then you have the metaproteome, where they're making the protein, and generally we believe the microbe is doing this because proteins are expensive. You have these amino acids you're chaining together. This is going to be expensive if it doesn't happen. But we don't know 100% that it is going to happen, or really what comes of it, because a protein can function many ways. So then you have the metametabolome or metabolomics. We actually can look at these substrates after the interaction and really see, here was the potential, here's how they're working on it, here's actually what came out of it. That gives us the whole picture. It's really hard for labs to do all this, not just financially, but if you think about the amount of time. As you know, bioinformatics is a bottleneck. Not a lot of people know about it. Not a lot of people have access to it. It can be expensive. It can be hard to request hours. It is challenging to get your mindset thinking about that. And then what makes it more difficult is we're training scientists either in bench or wet lab, as I call it, or in bioinformatics. So you become an expertise in one and not the other. I have challenged myself, which may have not been the best thing, but I've thoroughly enjoyed it, to have the whole picture, to be the jack of all trades. I made sure I was in the field sampling so I could see the environment, make the connection, and know the limitations of my work. I was in the wet lab optimizing the methods, getting the DNA, looking at it, knowing the limitations of my work. I then took the time to process it on the computer. Because of this, it takes so long to publish. and you can get scooped, or you're already in the dark. There are probably people watching this going, oh man, I have a metagenome, I thought it was the best, and now I found out it's not enough. I have to have all this stuff, I should give up. No, don't give up, right? It's keep pushing on it. I'm showing you where the field's going to make sure that we're trying to connect the whole picture. And this can be alleviated by trying to work with other scientists and their expertise. And then that brings me to the final part, is the expertise. While if you have the wet lab or you have the bioinformatics, you then have to stay on top of the tools and using the best methods and talking with others. And this can be so challenging. And then I guess the last thing I'll end with is nanosims, which is not necessarily a newer thing. It's not as used and it's more unheard of, just like stable isotope probing, not only because it's expensive, but also for the nanosims, it's hard to learn. There's only so many in the world. We happen to be a facility that has one. And the government in the United States, to try to help this, is they have these calls to have the national labs that have these capabilities, like JGI for sequencing, PNNL for metabolomics in general, and mass spec, and then us for isotope, radioisotope, and nanosims, to work with universities and other individuals to offer these services. So I want to say, if you're out there and you're wanting to use these, it's harder if you're not a US citizen, but it's not impossible. You got to look at connecting with labs or universities that have these capabilities to help push you to the future. Sorry, that's a lot.
Yeah, but it's thoroughly very interesting and it's an eye-opener to learn from the experiences that you have been through. And I really can connect with the statement that you made, that how you have gone through the training of the different aspects of being a life science researcher, and you have, although it takes a lot of time, you have gained the experience, and that has given you an understanding of the complete picture, which is, I think, very much essential. Well, people have been thoroughly enjoying your talk, and I can see that they are extremely glad that you shared so many things. Many people are agreeing to you, although there are a lot of questions from many of our participants, because it's something which is completely new. But since we have a time limit, and I know that you are also having a very busy schedule, we have to move forward with today's session. But it would be great that if you could share some ways to stay in touch with you, or maybe to read your publications, through which our audience can get to know your research in much detail, maybe your Google Scholar page or something, so that we can stay in touch and also learn from your publications that has been already published, and also the ones that are upcoming in the future.
I do want to say that I'm happy if you want to share my email. I might have a lot of emails from people, so it'd be nice if we could group like questions to me and I can try to answer them via email. Or if anyone's interested in opportunities. It's harder if you're not a US citizen. I'm sorry about that, but there's other ways you can work with your government, and there's governments that work with the US as well. For my publications, I do want to say I made it a point from the start that it's frustrating going through school, and when you try to research knowledge and then there's a paywall that just stops you. And it's frustrating, especially for the US, because a lot of our work's paid for with taxpayer dollars, but then we try to charge the public to get it. I mean, this is a worldwide problem. So I've made it a point to try to publish everything open access, and I'm pretty sure all except for maybe one or two of my publications are open access. So you should be able to access any of my data. And if you can't, reach out to me, because I think, there is one, maybe two, that if you reach out to me, I'm happy to share with you.
All right. Thank you so much, Dr. Gary, for taking out your time from your schedule. It was an amazing experience for all of us to listen to you, to learn from you. And we all hope that we can again get you back for some other sessions and learn even further from you. Thank you so much for joining us today.
Thank you.
Well, so I would move on further with, we have another great presentation coming up, and this is from one of our student fellowship holder at Pine Biotech. So before moving on, I would like to tell you that we have been learning so many interesting things about metagenomics, and it's so fascinating even to me, and I hope to everybody. I can see that in the chat. So you can definitely stay in touch with Pine Biotech, our host community, and the Omics Logic Metagenomics program. So this is a mentor-guided asynchronous training program where you can learn the basics of metagenomics, get introduced to the concepts, learn about the various techniques that we were discussing today with Dr. Trubl, such as the various sequencing approaches, the mass spec approaches. You can learn about them. You can learn about the various functions of the microbiome and how they regulate everything in our body, starting from energy generation and metabolism. And these training programs are designed in such a way that it can integrate the hands-on aspect so that you can learn by doing. And this is achieved with the help of the AI guided cloud based server platform of Pine Biotech, which is known as Server T Bioinfo. And they have various components such as NGS data analysis and metagenomics analysis and multi-omics analysis, including machine learning. So in terms of the metagenomics course, you would get introduced to how to analyze the amplicons, and with the help of the data to pipeline, which is a very popular analysis pipeline, you would learn how to get started with metagenomics analysis. And I also saw a few questions regarding databases where you can find data which you can analyze, publicly available data. So you can also get introduced to the various databases such as Silver, Green Genes, NCBI, RDB, where you can find data sets of various metagenomes and microbial communities. And the whole point of doing these sort of training programs is to enable you to get started with your own research project, to define your own ideas, to bring together on the table, and to get started to probably start the start of something great. You can start working on a project, get helped by the mentors, and you can take that project to a conference, maybe get some feedbacks from experts such as Dr. Trubl, and push forward your research career journey. To talk about this aspect and how the experience completely is, we have Dilara with us. So if you're interested in this metagenomics program, I will be sharing the link in the chat in some time. You can also watch a video of a webinar that we had quite recently regarding this program to learn the structure and the expected outcomes. But here's the deal. So Dilara Dikan is a graduate in molecular biology and genetics from the Bakışehir University in Turkey. She has been a research fellow at Pine Biotech, and she has been a metagenomics enthusiast. So she was working with metagenomics datasets, learning about it, and then she moved on to apply it in her own project. She's also an astrobiology enthusiast, especially looking after the health of astronauts. So she analyzed how metagenomics can play a role and provide better healthcare for astronauts who are living on the International Space Station. So it's our pleasure to have Dilara with us today, and we are all here to learn about what her project deals with, what was her experience of going through the programs, the training courses at Pine Biotech's platform, and ultimately being a part of the research fellowship where she got the opportunity to work on her own project. So without any further ado, I would welcome Dilara to take away the stage. Hi Dilara, how are you doing?
Thank you. That was a great introduction. I'm doing well. I hope everyone's doing well too. So I will immediately share my screen. I'm not sure if you can see it. Can you see it now?
Not yet. Okay, it's coming up. Yeah.
Okay.
We can see it now. Cool.
So basically, as Subrajit said, I am quite interested in metagenomics, microbiology, and astronaut health, astrobiology, all of those things. And I'll give a short introduction on how I got interested in these things. And then I'll talk about my project that I conducted during my internship at Pine Biotech. So, and overall is that my research was based on the adverse effects of high LET radiation on the mouse gut microbiome, and consequently on the nervous system, causing psychological disorders like anxiety and depression. So why am I so interested in microbes? And it's because when we think of microbes, we usually think of pathogens, but that's changing now. We don't only think of microbes that make us ill, but we also think of them as a treatment now. We can help use these microbes using metagenomic studies to find unconventional treatments for diseases like cancer or atopic dermatitis. And also because it's obviously a core of astrobiology studies, and it's essential in understanding evolution because we were coexisting for all these years. So we cannot understand our own evolution without the evolution of microbes. And of course, as Dr. Gary and as his discussion touched upon it, in climate change and agriculture studies and finding renewable resources, microbes are invaluable. So I chose this topic specifically, astrohealth, because of the prospects of space travel and how they advance. Therefore, the research involving space travel increases proportionately. So among biology, biotech, and human research conducted on the ISS, the International Space Station, are studies on how humans and animals are affected by space travel, which will be important for us in the future. So it's a fast-paced and growing field, and my research specifically is on a space-type radiation called ionizing radiation, and more specifically, high LET, linear energy transfer radiation. High LET radiation can penetrate into microstructures and has enough energy to cause chemical change, which leads prominently to cell death. And according to the information collected by the ISS research, the effects of radiation exposure on human health are currently uncontrolled and will continue to be so for the foreseeable future. One promising factor, however, is that host microorganism interactions will be optimized, to say the least, by around 2028. So here's my methodology. I had three main stages, which was data exploration, statistical analysis, and then drawing conclusions. In the data exploration stage, I found my data set, which was on mouse gut microbiota from NCBI, and then using the T-bioinfo platform, I used the DATA2 pipeline and the phyloseq pipeline on the data set that I had, which was 16 as amplicons, to find paths that could lead me further into understanding the data. So then I conducted a NOVA and t-test to find if the observations I made during the phyloseq stage were correct. And then finally, I tried to connect these results to palpable therapy ideas, potential therapy ideas. So my data set was a total of 80 samples, and it was divided into two categories. One was based on radiation, going from 0.1.25 and 1 GY radiation, and the samples were collected 10 days post-radiation and 30 days post-radiation. And here is one of the outputs that I got from the phyloseq pipeline, which is NMDS, non-metric multidimensional scaling, which helps us observe the whole data set and find relative connections between them or separations. And in this one, we can see the relative separations between the families of bacteria found in those samples collected from the gut microbiota. And here I observed from this angle that 10 days post-radiation is more grouped than 30 days post-radiation. And then another output of the phyloseq pipeline was the Shannon index graph. And the Shannon index is a value used for measuring the richness and evenness of species in different samples. So from my comparison here, I found that between the four different radiation doses, the Shannon index decreases with exposure to radiation at lower doses than at higher doses. And I also, during my research, found out that this was due to the properties of high LET radiation. And this is an excellent example that summarizes my entire findings during the data exploration stage. And from here, we can see from the phylogenetic tree that the OTUs that are significant, that change significantly from the control group, which was 0 GY. And we can see that 0.1 GY had lower abundances compared to 0. We can also see that OTU 4 and 5 have a common ancestor in recent times. So this is one of the most useful graphs that came out of the phyloseq pipeline that helped me make analytical observations. And then I decided to use statistical analysis, which was the ANOVA and t-test, to make sure that my observations were correct. And you can see from the simplified heat map that OTU 3, 4, 5, and 8 were the significantly decreased ones. And I also came to the conclusion that 0.1 GUI collected at 10 days post-radiation was a significant group. And I also found out that there was a connection between the samples collected at 10 days post-radiation and the radiation dosages. So there was no relation between those two categories. And the most significantly decreased ones were the ones that were exposed to 0.1 GUI radiation. So from this, I also came to the conclusion that the abundances for Lactobacillus, Bifidobacterium, and Erysipelotrichobacteria were the most significantly decreased abundances. The ones I'll be focusing on, however, is Lactobacillus and Bifidobacterium, since they contributed to the imbalance in the nervous system through protein coding genes that they have. And these protein coding genes correspond to GABA. And GABA is gamma aminobutyric acid, which is the chief inhibitory neurotransmitter in the central nervous system. And it is generated by Bifidobacterium and Lactobacillus too. Therefore, a decrease in these species of bacteria can cause or enhance disorders like depression and anxiety, which are very common in astronauts. And here you can see that there are two pathways, the GS and the GLS, through which these bacteria affect GABA. So from all of this, we can conclude by saying that lower doses of high LET radiation have a greater effect on OTU abundance. In this case, it is 0.1 GUI. And we can also surmise that the acclimatization period is between 10 and 30 days, since the samples collected 30 days post-radiation did not pass the significance threshold. But we also have to keep in mind that these mice were not exposed to radiation in the long term, the way astronauts are. And some of my limitations were, of course, the difficulty of the taxonomic profiling, since 16S regions are quite similar for bacteria in the same genus. And then there's also the technicalities of optimization, and the limited sample size was another issue. But all in all, this was a good precursor study for microbiota studies in space. And further research should include synergistic effect of radiation and microgravity together. And of course, in this whole stage, I don't know, because I didn't have a bioinformatics background, I don't know how I could have achieved this if I hadn't come across Pine Biotech and the T-Bioinfo platform, because that greatly helped me, along with the advisors like Dr. Mohit, Ilya, and Dr. Harpreet, who helped me throughout my research. So here are my references, and thank you for listening to me. I hope this was enjoyable.
Thank you so much, Dilara. That was an amazing presentation, and it's a pleasure to listen to you every time. Well, there would be one question that we have in the chat, and it would be great if you could address it in brief. For example, first of all, what was the duration of the study, the samples that were exposed to the radiation? What was the duration? Mm-hmm. And finally, if you could explain the results again, and their like the phylogenetics that you got, and what those families usually you can conclude from those families that you could find.
Yeah, so basically the duration was not specifically mentioned in the article that I chose the samples from, but it mentioned that it was during 1 day. So it wasn't as long-term as astronauts do. And we have to consider in animal studies, of course, like, you know, we can use mice, musculus, as a sample animal model, but we cannot expose it to as much radiation as humans are exposed to in space also. And I guess this was the phylogenetic one that you would like me to explain. So basically, when you run your samples through the phyloseq and data2 pipeline, They go through all the usual stages like normalization, and your samples are cleaned, etc. And then this is the output that you get. And from this, you can here understand that 1.1, 0.25, and 0. I don't know if I enlarge it, if you can see it more clearly. But from this, you can tell the abundances of OTUs, and OTUs are operational taxonomic units. When you run the data2 pipeline and the phyloseq pipeline together, you don't exactly get all of the genus and species names of the bacteria that are in the sample. First of all, because there are a lot of DNA samples that are not titled, that are not named. And then there are also the fact that when you run these DNA samples are very volatile and they break a lot. So estimations have to be made, and the system basically, when you run the pipeline, will align the DNA samples that are present in these microbiota fecal samples as much as possible, and then it will not give it a name. It will not, first, it won't run it through the databases and say like, this is lactobacillus or this is bifidobacterium. The first thing you get is operational taxonomic unit, which is similar to saying this is one type of bacteria, but it's not specifically named. So that's what OTU means here. That's what the OTUs are. And then what you get is these correspondences. OTU4 is, and OTU5 is bifidobacterium genus, and etc. And you don't usually get down to the species name if you don't optimize it too much, because of the limitations, as I said, of the numerous types of bacteria that are still not named or sampled. They're not in the database of these DNA samples, so they're usually left down to the genus name. So that's why that's how I made the correlations between OTU and the bifidobacterium and lactobacillus. Basically, I took out what was in the phyloseq pipeline, what this gave me as a correspondence. It gave me bifidobacterium and lactobacillus, and then I ran it again on the JGI database, and I found the genus and species name together, which are these ones.
That's great.
And phylogeny.
Yeah, and is there a correlation between the abundances that you found and the neurological activity with GABA that you mentioned?
Yeah, so basically, because I was using samples that were previously, collected by other researchers, this was like a totally bioinformatics study, I couldn't exactly go back and analyze the behavior of mice. So I couldn't conclusively say that the decrease affects the nervous system. I can't say that because I didn't work with those mice, basically. But what I can do is run through the JGI pipeline and find that these bacteria have protein coding genes for sure that affect the human nervous system. They basically through the central nervous system and through the interdendritic nerve, which is a nerve that goes through your gut and collects these chemicals, and it affects your central nervous system in return. So theoretically, I can say that a decrease in the abundances will cause anxiety and depression because GABA conclusively decreases. So that's what I theorize, basically.
Right, that's clear for us now. Thank you so much, Dilara, for your presentation. It was amazing to go through your project and to learn about the aspect where you can apply metagenomics in such a newly discovered field of science, which is up and coming. And this is something really exciting. Well, we also have many questions in the chat, so you can take some time to maybe address them in the chat, or maybe share some relevant resource which people can go through. And it's amazing to see such interest in the audience today, because that's the entire goal of organizing these webinars, where we can infuse the interest and excitement that we share as a community, and that's really amazing to see. So it's good to have so many different sorts of questions and to have that enthusiasm, because that is something which we always are on the lookout to address as a part of the various training programs running all across the globe as a part of Pine Biotech. So before we end today's amazing session, which has been such a great experience for all of us together, I would like to bring to the notice of your audience that about some of our great collaborations and partners with whom Pine Biotech has partnered to bring to you some exciting opportunities. So first of all, beginning with the people in the United States of America, Pine Biotech has recently partnered with NAPI, which stands for National Association of Idea Principal Investigators, which exists in 23 states in the United States of America and Puerto Rico. And the people who are residing in these states would get an opportunity to get access to the various training programs and the courses that Pine Biotech has designed. And you also get to work on the various project-based training programs that we have with mentor support. So that's something which you should definitely look forward to and keep an eye on. Pine Biotech's home ground is Louisiana, and therefore the partnership and the help that we have got throughout the years from Louisiana State University has been amazing. And our relationship is so great that now we have collaborated together to bring bioinformatics, machine learning in biomedical data science, transcriptomics, and various other domains of omics and bioinformatics courses and education to the people living in Louisiana at a much privileged manner. So if you are from the states of Louisiana and you know people, and definitely it's for many other people who reside in any part of the world can access now that we are offering it online remotely, so it gives you an added benefit. There has been recently a great collaboration with our international partners, namely Amity University, the Uttar Pradesh campus, and there was an MOU signing which has led to the massive collaboration between Amity University and Pine Biotech. As a part of this partnership and collaboration, we can now bring industry academic internships for people in India, students of any level, especially if you belong to the Amity University network and alumni group, you can get this access to the various industry academic internships and opportunities presented to you by Pine Biotech. But that's not the end. If you belong to any part of the entire Asian continent, this is good news for you, because Pine Biotech has also partnered with FABA, that is a Federation of Asian Biotechnology Association. They are a leader in various bioinformatics and biotechnological industries in the entire Asian subcontinent. As a result of this partnership, Pine Biotech can now give you a greater amount of opportunities to get training, advanced internships, projects, and even career opportunities if you are a part of our community and courses. So it's definitely a good option to stay connected with Pine Biotech and to be a part of all our upcoming workshops and opportunities. So just like we are having the Omics Logic Metagenomics training program running in the month of January, we are also going to launch a very exciting training program which is known as Omics Logic Genomics, that really goes deep into the domain of analyzing and interpreting genomic data, giving you the concepts of interpreting NGS datasets and leveraging programming languages such as Python and R to get the best out of your dataset and to understand the biological reasoning behind the datasets that are produced. Now, I would also like to inform you about some exciting workshops coming up very quickly. So this is on 11th and the 12th of January. This is a part of the Omics Logic Genomics program, and this workshop will introduce you to the concepts of genomics and its application in biomedical data science. We will also touch upon genomics and NGS data analysis, something which I was telling you right just now, pre-processing data analysis, genomics in R and Python. There will be a guest lecture by Dr. Sanjeev Kumar, who is the founding director of BioCos Life Sciences and alumni of Indian Institute of Science, Bangalore, who is an industry expert in programming, multi-omics, machine learning, deep learning, and also a research mentor. And he will be sharing his insights on single-cell genomics and high-throughput spatial genomics. There will be many other exciting talks on NGS single cell data analysis and various project examples that students have done and also you can be a part of in the future. And we will also touch upon how genomics can really improve the infrastructure and quality of agriculture in our world, such as Dr. Trubl was touching upon the impact these microbes can have on our food, and to analyze that and to really increase the yield of our production, definitely we need to leverage genomics and metagenomics. So that is something which you can learn by being a part of this workshop. So here's the complete schedule of the 2-day workshop. I would request you to take screenshots of this particular slide if you're interested in checking out any of the sessions. I would recommend you attending all the sessions to get the most out of these opportunities. And the best way to keep in touch with us and to know about any of the upcoming opportunities and events that we have here, you should definitely stay in touch with our social media handles. I invite you all to check out the links in the chat. You will find that we are present in every platform out there. And I also invite you to join us in the various groups that we have on Facebook and LinkedIn, because the members of this group are constantly communicating with one another. You would find various students who are at your level and various experts who are there who have the best knowledge of the art that we are doing. So it's really a great opportunity for you to interact with them and to learn about various opportunities and options. So I highly recommend all of you to check out the links and join us in this particular journey. Now, Sci-Omics is a recurring event. It's not a one-stand event. So we had some great people who shared their research and insights in the month of November on bioinformatics and data science. In December, we did a really interesting and exciting session on study abroad, because that was a popular interest amongst various students who wanted to learn about various scholarship opportunities, how applications need to be made, what's the best design of a CV, etc. So we gathered people from all around the globe who has been through this process to talk about this thing. Now it's the month of January and we are focused on metagenomics. Just like an amazing session that we had today, we are also having other sessions coming up, which I will give you the insights on really quickly. In the month of February, we will have sessions on bioinformatics and data science that would touch upon the role of various machine learning models and various algorithms that you can leverage in your project. And all of this leads up to the mega event that we have in the month of March. That's the Omics Logic Research Symposium, our conference, which will be held remotely so that everyone can join in. And there will be some great talks by some great scientists from all over the world, both from academic and industry. And there will be also an opportunity for students to showcase their research as a part of the poster presentation session. So check out our social media handles again. That's the reason I shared them with you. Very soon we will be rolling out the various call for abstracts where you can submit your research project abstracts and participate in the poster presentation competition. So do keep an eye on that call for abstracts for you guys. So talking about the upcoming sessions, the next Sci-Omics sessions when we will meet again is on the 21st of January. It's a Friday again, and the time is 3 p.m. CST, 9 p.m. GMT. And this session will be done by Dr. Christopher M. Taylor, who is Associate Professor at the Department of Microbiology, Immunology, and Parasitology at the Louisiana State University Health Sciences Center. And he will be talking about metagenomics and microbial communities. He has a rich experience of training students in metagenomics and making them ready for analyzing any metagenomics data. So I'm looking forward to seeing all of you on the 21st of January for this exciting session. And I hope that you can make the best out of all of these webinars that we organize. In the month of February, as I said, we will be having Dr. Abhishek Sengupta, who is an assistant professor at the Center for Computational Biology and Bioinformatics at the Amity Institute of Biotechnology in Amity University, Uttar Pradesh. And he will be talking about network construction and inference as an elegant approach to understand disease pathogenesis, which is a very trending topic in the times of COVID-19 and other pandemics. So the date is 4th of February 2022 and the time is 5 p.m. IST, that is Indian Standard Time. So again, keep an eye on our social media handles. We will be rolling out all these notices again there along with the links. So finally, it's a request to all of you at the end of this brilliant session to fill this feedback form, because that is essential for us to make sure that we deliver the best for you people. And it's a request to please go and fill out this form. It won't take more than 2 minutes, but it will help us provide you the best experience in the future. So I hope that you had an amazing session today, because definitely I had a lot of fun learning about so many amazing topics today. And it's just the beginning. 2022 will have many exciting sessions, which will be a very impactful addition in our lives as student researchers. So finally, I would end on the note with this note that join our Sci-Omics group. We are there on various social media channels, such as we have these groups on WhatsApp, Telegram, and Slack. You can check out our website as well, and you can scan these QR code to be redirected to the website, and you can get all the information out there. So on that note, it's time to bid all of you goodbye. It was a lovely experience to talk to all of you and to see this wide interest, and I hope that we can only grow from this particular point in the future. Thank you everyone for joining us today, and wish you all the best with your future. May 2022 be extremely productive and fruitful and happy for all of you. Thank you so much everyone for joining, and see you soon.